Cellular exchanges under isotonic, hypotonic, and hypertonic conditions. 1. Under isotonic condition. An isotonic solution is a solution that has the same osmotic concentration as the cell that is placed in it. Example: blood plasma is isotonic to the body cells. When red blood cells are put inside an isotonic solution, the cells remain the same because water gained by the cells is equal to water lost. 2. Under hypotonic condition. A hypotonic solution is one that has more water molecules and less solute. When a cell is placed inside a hypotonic solution, it swells up because more water enters it than leaves it. The cell becomes turgid. Some cells can burst if there are no special mechanisms to remove water. Example: plant cells have the cellulose cell wall which prevents excessive swelling. Red blood cells without cell wall will burst if placed in a hypotonic solution. This process is called hemolysis. 3. Under hypertonic condition. A hypertonic solution is one that has higher concentration of solutes and less concentration of water than the cell that is placed in it. When a cell is placed in such a solution, the cell will lose water by osmosis into the hypertonic solution. The cell will shrink and even die if the process continues. If a red blood cell is placed in a hypertonic solution, shrinking occurs, and this is known as crenation. Turgidity and flaccidity. The plant cell has a definite shape due to the presence of a cellulose cell wall. The cell wall encloses a concentrated cell sap. The cell wall does not play any important role in osmosis, but the cell membrane and the cell sap are very important. If a plant cell is placed in a hypotonic solution, example distilled water, water will enter the cell sap by osmosis through the cell membrane. The cell swells up and the cell membrane is pushed against the cell wall with a pressure called Turgot pressure. The Turgot pressure is the pressure caused by the cell constituents on the cell wall as the cell takes in water by osmosis. The cell wall on its part tries to resist or stop expansion of the cell sap by pushing with an equal and opposite pressure called wall pressure. When a cell is placed in a hypertonic solution, example salt solution, it loses water continuously through the cell membrane. The volume or the size of the cell sap reduces and the cell shrinks. The cell is said to be flaccid. The cell membrane starts to be pulled from the cell wall and the cell becomes weak. When the flaccid cell continues to lose water by osmosis, the protoplasm shrinks and the cell membrane pulls from the cell wall completely. The large central vacuole forms a small ball at the center of the cell. The cell is now said to be plasmolysed. Plasmolysis is easily reversible by placing the plasmolysed cell in a dilute or a hypotonic solution like distilled water, or else the cell becomes completely damaged. If a plant is exposed to conditions that plasmolysis occurs, the stem becomes weak and the leaves collapse or fall off. This process is known as wilting. Wilting prevents the excessive loss of water by plants during the dry season. Wilting is the loss of turgidity in a plant due to excessive transpiration.